Have you ever wondered how your body fights off everything from a common cold to maybe a more serious infection?
Mm-hmm.
Or perhaps how it somehow remembers past invaders so it can respond even faster next time?
It's quite amazing when you think about it.
It really is remarkable, isn't it? Our bodies are, well, constantly battling behind the scenes, deploying this sort of invisible army to defend us.
Absolutely. A silent defense force.
Today, our mission on the deep dive is to unpack 3 absolutely crucial components of that internal defense system.
Okay.
We're going to explore cytokines, the primary and secondary immune responses, and the intricate complement system.
3 really key areas.
Exactly. By the end of our conversation, you'll have a much clearer, more insightful picture of these unsung heroes, how they work together, and their surprising real-world impact.
Yeah, it's a good shortcut to understanding some pretty complex biology.
It really is.
That's right. And what's truly fascinating here is that these aren't just, isolated functions.
Right.
Think of them less as individual players and more like a finely tuned orchestra, each section playing a vital role in keeping us healthy and protected.
I like that analogy. An orchestra.
Yeah. Our goal today is to dive into not just what these complex biological processes are, but maybe more importantly, why they function the way they do.
And why that matters for your health, ultimately.
Precisely, the relevance.
Okay, let's unpack this first piece then. Imagine your immune system as, like, a vast, bustling city.
Okay.
Different departments constantly needing to communicate vital information, and fast. How do they manage that?
Good question.
Well, that's where cytokines come in. They're kind of the immune system's secret messaging service.
That's a perfect analogy, actually. Cytokines are best described as small, hormone-like proteins. They act as vital intercellular messengers.
So tiny communicators.
Tiny but powerful. They're secreted by a really wide variety of cells throughout your body, not just the classic immune cells like lymphocytes and macrophages,
Oh, really? What else?
Also, cells in your skin, blood vessels, even platelets can produce them.
Huh. Interesting.
Yeah, and they're typically produced on demand. So in response to triggers like infection, toxins, or inflammation, they essentially sound an alarm or give instructions.
Like sending out an urgent memo?
Exactly. And you'll sometimes hear them referred to by different names, like interleukins, for instance. That just means they communicate between different white blood cells.
Got it. So if there are these critical messengers, what are their unique personality traits? What makes them so effective and, well, versatile?
Well, they have several fascinating characteristics. First, there's something called pleiotropy.
Pleiotropy.
Yeah, it means one single cytokine can have multiple different effects. Like one command from headquarters triggers several distinct actions across different units.
One messenger, many jobs. Okay.
Exactly. Then there's redundancy. The same job or activity might actually be triggered by several different cytokines.
Backup system.
It's a brilliant built-in backup system, yeah. Ensures that if one messenger is compromised, another can still deliver the same crucial instruction.
Makes sense.
They're also incredibly potent, active even at extremely low concentrations. A tiny whisper can create a huge impact.
Wow.
And as I mentioned, they're generally not stored. They're synthesized rapidly as needed. An on-demand response.
So efficient.
Very. And finally, their modes of action vary. They can act on the cell that produced them, that's autocrine.
On itself.
Or on nearby cells, paracrine. Or they can travel through the bloodstream to distant target cells, that's endocrine action.
So local memos, interoffice comms, or system-wide alerts.
You got it. Great way to put it.
Okay.
So when we look at their functions, it's clear they are absolutely fundamental. They initiate and regulate pretty much every stage of an immune response.
Every stage.
Essentially, yes.
Oh.
They orchestrate everything from guiding immune cells as they develop, differentiating, right up to directing the full-blown attack on invaders. For example, in inflammation, they are major mediators. They amplify the process, increase vascular permeability.
Letting more immune cells get to the trouble spot.
Exactly. They also drive cell activation and proliferation, basically telling various immune cells to wake up, multiply, and get to work.
Mobilizing the troops.
Precisely. Beyond that, they're crucial for things like antibody production. Specific ones like IL-4 and IL-10 are key for activating B cells to produce and even refine antibodies.
Right.
For antiviral defense, you have interferons. They're your body's frontline response, inhibiting viral replication and activating other cells like macrophages and NK cells to clear infections.
Interferons. Okay. Heard of those?
Yeah, they're quite famous. Cytokines even manage leukocyte trafficking.
Traffic controllers for immune cells.
Sort of. Using a special subset called chemokines, they direct these cells to sites of infection. Think of them like chemical breadcrumbs guiding the immune army.
Okay, that's a cool visual.
Isn't it? And some cytokines, like interferon gamma, are essential for macrophage activation, turning these sort of scavenger cells into potent microbial killers.
Powering them up.
And yes, they even cause fever. Things like IL-1 and TNF-alpha act as endogenous pyrogens.
So they're why you get a temperature when you're sick.
They're a big part of it, yes. Raising your body's temperature makes it less hospitable for pathogens. They also assist in tissue repair and wound healing.
So not just fighting, but rebuilding too.
That's right. And they're involved in aspects of tumor immunity, helping your body identify and eliminate cancerous cells.
Wow, the list keeps going.
It does. And finally, some, like IL-10, are actually crucial for immune suppression and tolerance.
Suppression? Why would you want that?
It's essential. They ensure your immune system knows when to stand down, when the fight's over, and importantly, doesn't attack your own healthy tissues. Autoimmunity prevention.
Ah, okay. The brakes.
The brakes, exactly. So, you see, far from being just simple signals, cytokines are the immune system's sophisticated orchestrators.
Directing the whole symphony.
Right. Directing defense, inflammation, healing, often with surprising overlaps in their roles, which points back to that redundancy we talked about. The system is built to rarely fail.
That truly is complex, almost mind-boggling, the number of jobs for such tiny messengers.
It is.
So given how fundamental they are, how has understanding cytokines translated into, well, real-world applications? Clinical significance for our health.
Oh, the insight we've gained has been huge therapeutically. For instance, we now use recombinant cytokines, lab-made ones, to treat infections, especially in immunocompromised patients.
Boosting their defenses.
Exactly, helping their systems fight back. They're also proving vital in modulating immune responses in autoimmune conditions and allergies where the immune system might be, overreacting or misfiring.
And rebalance it.
Precisely. And in cancer treatment, some cytokines are used to boost the immune response against tumors, or to stimulate the production of crucial immune cells after therapies like chemotherapy, which helps prevent infections.
Makes sense. Chemo can knock down your Y cells.
It can, yeah. So things like GCSF help rebuild them. However, it's a critical balance.
There's always a catch.
Well, yeah. While beneficial, an excessive production of cytokines, sometimes called a cytokine storm, can lead to severe pathological conditions. Things like sepsis or septic shock.
Where the immune response itself causes the damage.
Exactly. Yeah. Widespread tissue damage. That's why we also have cytokine inhibitors, like certain steroids, which can dampen these runaway responses. It's all about fine-tuning this incredibly powerful messaging system.
Delicate balance indeed. Okay, now, here's where it gets truly fascinating for me. How does your body learn from past invaders?
Immunological memory.
Yeah. Think of it like a detective building a case file. The first time, it's maybe a slow, meticulous investigation. But the next time that same suspect shows up, bang. The response is much, much faster because the detective immediately recognizes them.
That's precisely what happens with our adaptive immunity. The primary response occurs the very first time your body encounters a specific foreign antigen, a new threat it hasn't seen before.
The first meeting.
Right. There's an initial lag phase where, no antibodies are detectable yet. The immune system is busy identifying the threat, activating the right cells, preparing its defense.
Gearing up.
Exactly. This period varies, but during it, a small clone of specific B cells and plasma cells begins to form. Initially, you typically see IgM antibodies first,
IgF,
followed by an increasing proportion of IgG. The overall antibody levels are generally lower in this primary response, and they rise steadily over days to weeks before eventually declining. It's a necessary, but somewhat slow, first punch.
Learning the ropes, essentially. So if that's the learning phase, what happens the next time you meet that exact same antigen? Does it truly remember?
Oh, absolutely.
And does that memory make the fight different?
Dramatically different. This is where the secondary immune response showcases the brilliance of our body's memory system.
Okay, I'm ready.
Upon re-exposure to that same antigen, even if it's years later, the response is remarkably rapid, robust, and incredibly efficient.
Faster, stronger.
Much faster, much stronger. The lag phase is dramatically shorter, often just a few days instead of a week or more.
Wow.
The level of antibody produced is much, much higher, sometimes 10 times greater, or even more than in the primary response.
10 times, yeah.
And crucially, these antibodies are predominantly IgG, which tend to persist longer and are often more effective in various ways.
IgG takes over from IgM mostly.
In many cases, yes, it becomes the dominant player. And another key thing, these antibodies produced during the secondary response generally have a higher affinity.
Higher affinity, meaning?
They bind to the antigen much more firmly, more tightly. This happens because of a process called somatic hypermutation during the B cell development. Think of it like the immune system refining its weapon between battles, making it a perfect, high-precision match for that specific invader.
So it's not just faster, it's better.
Exactly. It's not just about fighting. It's about learning and improving. Your immune system is a master strategist. It takes its first punch, maybe a bit slowly, to gather intelligence. So the second time, it's not just faster, but smarter, more precise, and devastatingly effective. It's a true evolutionary marvel.
And this is all thanks to memory cells.
Precisely. This accelerated and enhanced response is thanks to long-lived antigen-specific memory cells, both B cells and T cells, that were generated during that initial primary encounter.
They hang around.
They do. They require less stimulation to get going, and they proliferate rapidly and in large numbers when they see their target again.
Which explains vaccines.
Perfectly. This is precisely why vaccinations are so incredibly effective. They strategically prime your body with a safe version of the primary response.
Setting up those memory cells.
Exactly. Setting you up for a powerful, fast secondary defense if you ever encounter the real threat.
Brilliant. Okay, so we've talked about communication with cytokines and learning and memory with primary and secondary responses.
Right.
But beyond these incredible systems, how does the immune system, directly attack and clear out threats? What are the weapons?
Okay. Now we're getting into another major player, the complement system.
A complement. Sounds helpful.
It is. Think of it as a kind of immune alarm and a weapon, all rolled into one complex system.
Alarm and weapon.
The complement system is indeed a formidable and vital part of our immune defense. It's a complex network of proteins, maybe about 20 to over 30 of them, mostly found dissolved in your blood serum.
Run around.
Yep, acting as a powerful amplification cascade. These proteins normally exist in an inactive state, like proenzymes, just waiting to be triggered.
Dormant until needed.
Exactly. And they make up a surprisingly significant portion of our total serum proteins, nearly 10% sometimes, which really highlights their importance in defense.
Wow, that much.
It's found in pretty much all mammals and even many lower animals, which tells you it's evolutionarily quite old and important.
Okay. Any key properties?
A key one, historically important, is its heat lability. If you heat serum to 56 degrees Celsius for 30 minutes, you destroy its activity. Labs used to do this deliberately to inactivate complement.
Interesting bit of history.
Yeah, and these proteins are primarily synthesized by your liver cells, though other cells like macrophages contribute too. And production ramps up when they're actively being used, when they're being consumed in an immune reaction.
So the body replaces them quickly.
It tries to, and typically they get activated when they bind to pathogen surfaces or often to antibody-antigen complexes. They don't usually bind to free antibodies or free antigens.
The exact complex.
Generally, for the main pathway, yes. It's crucial for both our innate, immediate defenses and our acquired specific immunity. It bridges both arms.
Okay. It sounds like a powerful, dormant system just waiting for a signal. So how does this incredible system get activated? How does it spring into action?
Well, it activates through three main pathways. Think of them as different triggers, but they all converge on a central step, the activation of a key component called C3.
C3 is the linchpin.
C3 is absolutely central. Its activation is key, and ultimately all pathways lead towards forming what's known as the membrane attack complex, or MACE.
Membrane attack complex. Sounds serious.
It is. Okay, so the pathways. First, we have the classical pathway. This is the one primarily triggered when antibodies, specifically certain types like IgG and IgM, bind to an antigen, usually on a pathogen surface.
So antibodies are the main signal here.
For the classical pathway, yes. Antibodies binding essentially flags the target for complement. Interestingly, some other non-antibody molecules can sometimes kick it off too, but antibodies are the classic trigger.
Got it. What's next?
Second, there's the alternative pathway. Now, this one is particularly important because it doesn't strictly require antibodies.
No antibodies needed.
Not necessarily. It can be stimulated directly by the surfaces of microorganisms themselves, like certain components of bacterial walls, such as endotoxins.
So it can react immediately to bugs.
Exactly. This makes it a crucial early responder in an infection, active even before your body has had time to produce specific antibodies. It's a more direct route to activation, part of the innate response.
Very useful early on. And the third?
The third is the lectin pathway. This one is initiated by lectins, a type of protein our body makes, specifically one called mannose-binding lectin, or MBL.
MBL, what does it do?
MBL binds to specific sugar molecules, like mannose, which are often found on the surfaces of bacteria, fungi, and viruses, but importantly are less common on our own healthy cells.
So it recognizes foreign patterns.
Precisely. Once MBL binds to these sugars on a pathogen, it acts in a way very similar to the first part of the classical pathway, activating other complement proteins and leading, again, to C3 activation.
So 3 different ways to get to C3.
Right. 3 different initial triggers, antibodies, microbial surfaces directly, or microbial sugars, but they all converge to activate C3 and unleash the power of the system. It ensures invaders have fewer places to hide.
Okay, so it gets activated through these different routes. Classical, alternative, lectin. But what does it do then? What's the actual punch it delivers?
Right, the effector functions. They are profound and multifaceted. Perhaps its most dramatic action is lysis, which means cell bursting.
Bursting the invader.
Exactly. The final stage of complement activation forms that membrane attack complex, my LAC we mentioned. It's essentially a molecular drill or pore.
A pore.
Yeah, the C5b9 complex inserts itself into the target cell membrane, like punching a hole in it. This causes water and salts to rush in uncontrollably, making the cell swell up and burst, pop.
A direct kill mechanism.
A very direct hit. It's particularly effective against things like gram-negative bacteria, which have thinner outer membranes. Gram-positive bacteria with their thick cell walls are often more resistant to this direct lysis, though.
Okay, so lysis is one punch. What else?
Another crucial function, arguably its most important overall, is opsonization.
Opsonization. Heard that word. It means making things tastier for phagocytes.
That's a great way to think about it. Essentially, complement components, primarily one called C3b, bind all over the surface of microorganisms or cellular debris. They coat it.
Marking the target.
Exactly, like putting a big, glowing "Eat Me" sign on the invader. Your immune scavenger cells, the phagocytes like macrophages and neutrophils, have specific receptors on their surface that recognize these complement-coated particles.
So they grab onto the C3b.
Precisely. This significantly enhances their ability to engulf and destroy the pathogen. It's called facilitated phagocytosis. It makes the whole process much, much more efficient.
Okay, coding for destruction. Very important.
Absolutely vital. Then, complement fragments also act as major mediators of inflammation.
How so?
Well, smaller fragments produced during the activation cascade, like C3a, C4a, and especially C5a, are potent anaphylatoxins.
Anaphylatoxins. Sounds like anaphylaxis.
Related, yes. They rapidly trigger inflammatory responses. They stimulate cells like mast cells and basophils to release substances like histamine.
Histamine. Causes swelling, redness.
Exactly. Increases capillary permeability, causes smooth muscle contraction, edema, swelling, all hallmarks of inflammation, designed to help fight the infection. C5a is also a powerful chemotactic agent.
Chemotaxis. Attracting other cells.
Yes. It acts like a chemical beacon, a strong attractant signal, drawing phagocytes, those neutrophils and macrophages, out of the bloodstream and towards the site of complement activation, towards the battleground, calling in reinforcements.
So it recruits help to the area.
Aggressively recruits help. So you can see, complement isn't just a blunt weapon like lysis. It's also a sophisticated alarm system, inflammation, a targeting system, opsonization, and a recruitment system, chemotaxis.
Well, instinctual.
Very. And it even helps in other ways, like neutralizing viruses directly or helping clear out those immune complexes, the leftover antibody-antigen debris from battles transporting them to the liver and spleen for disposal by macrophages. It even enhances antibody production by B cells.
Wow. Okay, this system is incredibly powerful. Almost sounds like it could be dangerous if not controlled. With such immense power, how does the body keep this system from attacking its own healthy cells? There must be some really tight regulation, right?
You're absolutely right. Uncontrolled complement activation would be disastrous. So, to prevent unwarranted damage to our own host tissues, the system is under very tight regulation by various inhibitory substances.
Safety mechanisms.
Multiple safety mechanisms. For example, our own healthy cells express specific proteins on their surfaces, things with names like DA, decay accelerating factor, or MCP, membrane cofactor protein, or CD59.
What do they do?
They essentially act as off switches. They can quickly inactivate or dissociate components that accidentally land on our cells, basically telling the system, whoa, not here, this is a friendly cell, stand down.
Self-recognition signals.
Exactly. There are also soluble inhibitor proteins circulating in the blood, like C1 inhibitor, which prevents the classical pathway from kicking off inappropriately. Factor H and factor I regulate the alternative pathway. It's a multi-layered control system.
So checks and balances everywhere.
Checks and balances are crucial to ensure that this powerful weapon is primarily directed against genuine threats and not turned against our own healthy body.
Okay, that makes sense.
Yeah.
But what happens when these regulations fail, or if someone is missing a component?
That leads to what we call complement deficiencies. These are often inherited genetic conditions where a person is missing or has a defective version of one of the complement proteins or regulators.
And that causes problems.
It can cause significant problems, generally falling into two main categories. First, and perhaps most obviously, an increased susceptibility to infections.
Makes sense. Weaker defense.
Right. Especially recurrent infections by certain types of bacteria, particularly encapsulated bacteria. For example, deficiencies in components like C3 are linked to severe infections, while deficiencies in the later components, C5 through C8, which form the MAC, are strongly associated with recurrent infections by Neisseria bacteria, like those causing meningitis or gonorrhea.
Specific vulnerabilities.
Yes. And severe liver disease can also lead to acquired complement deficiency, since the liver makes most of these proteins.
Okay, so infections are one issue. What's the other?
The second major issue is an association with certain autoimmune diseases, like systemic lupus erythematosus, SLE.
Autoimmune. How does lacking complement lead to autoimmunity? Seems counterintuitive.
It does seem odd at first, but it relates to that clearance function we mentioned. Complement, especially the early components like C1, C2, and C4, plays a crucial role in clearing immune complexes from the body.
The leftover debris.
Exactly. If you can't clear these complexes efficiently because of a complement deficiency, they can accumulate in tissues like the kidneys or joints, trigger chronic inflammation, and contribute to autoimmune conditions like lupus.
So it's a failure of cleanup leading to bigger problems.
Precisely. And there are also conditions caused by defects in the regulators. A notable example is hereditary angioedema. This is caused by a deficiency in that C1 inhibitor protein we talked about.
The one that controls the classical pathway start.
Yes. Without enough C1 inhibitor, the complement system becomes sort of chronically partially active, leading to the overproduction of certain fragments that increase vascular permeability. This causes episodes of localized, sometimes severe swelling in tissues.
Wow. It really highlights the critical need for that balance.
It absolutely underscores the delicate balance required for this powerful system to work for us and not against us.
Wow. Okay. We've really journeyed through an incredible, intricate world today. From cytokines, the body's vital messengers,
The communicators,
to understanding how our immune system learns and remembers through those primary and secondary responses,
the memory system,
and finally exploring the formidable complement system, acting as both that alarm and a direct weapon.
a complex arsenal.
It's truly a testament to the quiet genius operating within our own biology, isn't it?
It really is. And I think what powerfully comes across is that these aren't isolated functions at all.
No, clearly interconnected.
Deeply interconnected systems. It illustrates the immense complexity and the delicate balance required for our immune system to effectively protect us. It's just this dynamic, adaptive, highly sophisticated defense network constantly working behind the scenes for your well-being.
So maybe next time you feel a bit under the weather and then, you bounce back.
Yeah.
Take a moment to consider that invisible army. The messengers, the memory cells, the molecular weapons.
Yeah.
They just won a battle for you.
A silent victory happening all the time.
Exactly. And maybe a final thought to leave our listeners with. Given the immense power and the intricate regulation of these immune components we've discussed, what new challenges or maybe opportunities might we discover as our understanding of how they all interact continues to deepen?
That's a great question. The more we understand the interplay, the more potential we unlock for targeted therapies, for better vaccines, maybe even ways to fine-tune the immune response itself. It's a frontier that's still very much being explored.
Lots more to learn.